Hello everyone, and welcome back to our Lecture 12 over Metabolism for our Microbiology lecture series. Let's go ahead and start on this. It's going to be a very quick run-through of metabolism. I'm going to hit the highlights. I'm going to rely a little bit on our secondary supplementary activities, our YouTube videos and things like that to walk through some of the really in-depth movements of some enzymes and things around inside of the metabolism process. So let's go ahead and start this up. So what is metabolism to start with? Well, metabolism is the chemicals and physical workings that go on inside of a cell. So all cells have chemical reactions going on inside of them all day long, hundreds of thousands of them all day long, all the time, constantly. If it's a physical thing like an enzyme changing or a chemical reaction breaking apart, like forming ATP or ATP breaking apart, releasing energy and things like that, all of those different reactions make up the sum of metabolism. So there are two different types of chemical reactions. You're probably familiar with these. You have reactions that build things up, anabolism, and reactions that break things down, catabolism. So anabolism builds things up from simpler to more complex. Catabolism breaks things down from more complex to simpler. These tend to take energy to do this, build things up. This tends to release the energy that's found within, taking things from more complex to simple. So enzymes is kind of a big deal here in our concept of metabolism. They play a huge role in metabolism. You're probably familiar with what enzymes are. They're a form of some sort of biological protein, a biological catalyst that helps reactions that would either be very difficult to achieve or very slow to achieve and things like that go faster or be easier to start. So it would be essentially like if I had to push my car up a hill, you can see this here, that would take a lot of energy for me to do so. But an enzyme would be essentially like having a winch that takes 80% of the actual work for me, so I just have to push a little bit of the weight of my car up the hill. And that's what an enzyme does. It makes things way easier for reactions to start. So you can see this here. This is the energy. So without an enzyme, it would take this amount of energy to start this reaction to make these particular things, take apart these things from 1 into 2. Or with an enzyme, you can see the energy is significantly lower. There's way less energy that's required to do that. So this is called the activation energy, the amount of energy that it takes to start a reaction, to get it from point A to point B, to start that process, the activation energy. And enzymes tend to lower the amount of activation energy that's required to make it easier on cells, so they don't have to sit there and work really hard all day long to do things that otherwise should be quite easy. It can also help by making reactions easier to occur. So if two things would be very hard to get them together, we kind of have float around and bump into one another in the cell. An enzyme can hold one of them and then the other one falls into the enzyme too, so you've got both of them really close together and then the enzyme can just shove them together kind of thing. So it makes things easier. Now, the cool part about enzymes is they're not broken down or changed at all in these reactions. So an enzyme can work once, it goes right back to the way it was. It can do another reaction instantaneously, and it can do billions of them because they never break down just naturally from use. Now they can break down from things like heat, detergents and chemicals and things like that, but they're not going to naturally break down just from use. So there's a couple of types of enzymes. They're pretty easy. You have a simple enzyme, which is just a ball of protein that does something. And you also have something called holoenzymes, or conjugated enzymes. Holoenzymes tends to be the more common term. This is going to be an enzyme and some sort of non-protein function that goes along with it. So what you'll see are things like Apoenzymes, which is the protein portion, and then the cofactor that goes along with it, which is the non-protein portion of this holoenzyme. Cofactors essentially are little extra bits that help enzymes work. So you can see down here we've got our apoenzymes here with the purple, and then you've got the coenzymes or the cofactors that are stuck on the side. These are essentially little aids. So a simple enzyme is just the purple. It works by itself. You don't need a cofactor or a coenzyme to make them work. Whereas a conjugated enzyme or a holoenzyme requires both the purple and the coenzyme or the cofactor to function, kind of like having to put your thumbprint on the little retina scanner before you can open the lock kind of thing. You have to have an extra step involved instead of just opening the door kind of thing. So cofactors are pretty cool, different types of enzymes. So you can read them here, what they do, what their different types of, you guys are probably familiar with the ACE ending. Anything that ends in ACE is usually going to be an enzyme, and that usually tells you what it breaks down, things like that. So we can see over here the cofactors that are required as well to break down certain things with those particular enzymes. So enzymes, you're probably familiar with the concept of folding of proteins, primary, secondary, and tertiary structure. All these different foldings of the structures and things like that are very important for enzymes to work. You're also probably familiar with that concept of an active site as well, where the enzyme is going to bind. You can see here our active site right there, active site right here, and when the enzymes are put together, you have two active sites, one on either side from the purple and the red. So you can see the active sites down here. Enzymes are very specific. Only certain substrates will fit inside those active sites, and they only work on certain things. Once again, this is really biology 1010 concept here. You're probably familiar with this stuff. So sometimes you can have something called a temporary enzyme, which is an enzyme that has something called an induced fit. It's going to be when a substrate that kind of works gets close enough to the enzyme, and another substrate is going to come in and be broken apart, and then it can kind of fit. You can kind of see it sort of works, kind of, sort of, but it works close enough that it can make the reaction occur anyway, which is kind of cool. Weird concept here. I'm going to skip over most of this on the exam. This is a really weird concept. So here's cofactors, what they really do. So here's our enzyme here. And a cofactor or a coenzyme is going to essentially kind of serve a role to hold something in place. So you can see here this little red S here, substrate number 1 or 2, wherever that is, the little red ball, is going to essentially be stuck to the green enzyme over here, the green part of our product. And you can see the cofactor here holds the little red ball, the little red substrate in place. Now, without that cofactor to hold, or the coenzyme to hold that little red ball in place, it's going to pick it up from substrate 1, rotate it around, and stick it into substrate 2, which is what we need to do. Now, without that little coenzyme there to do this, it would be very, very difficult, take a lot of energy for this enzyme to be able to form a structure that could hold a ball, and then rotate the ball, and then redeposit the ball. Whereas we can just have a little coenzyme that does it for us. Now, coenzymes are things like nickel, copper, you saw a bunch of them on that earlier chart, things that we eat. Little small amounts are needed for these micronutrients, little small amounts in your diet, because they serve such important roles in your enzymes. So this will be a little piece of nickel, a little ball of nickel, and some other couple of atoms put together here, and part of that cofactor, coenzyme, and things like that, to help your body carry out very specific reactions that are very crucial for you to survive. Now, there are two different types of enzymes on a really broad scale: exoenzymes and endoenzymes. Get the idea from the name, fairly easy here. Exoenzymes are going to be enzymes that are going to be secreted by an organism outside of their cell to break down really large molecules of food or harmful chemicals and things like that. So a couple of enzymes that you're probably familiar with here, exoenzymes, are cellulase, amylase, penicillinase. Amylase breaks down starch, penicillinase breaks down penicillin. This is how bacteria survive in the presence of an antibiotic. Cellulase breaks down cellulose, bacteria that eat that for cows and things like that in their stomachs. Those enzymes are quite large. They're going to be secreted outside of the organism's cell, break down all of the sugars, break down all of the bacteria, break down all the starch for the bacteria, break down all of the antibiotic in the environment. So the bacteria itself never actually has to take in penicillin. It gets the preformed broken down of cellulose, because cellulose is such a large molecule, it can break it down outside. Amylase breaks down starch. Once again, such a large molecule breaks it down into smaller bits outside of the cell, and then it can suck it in for nutrients, break it down in a smaller package, which is really useful. And then you have endoenzymes, which work the other way around. They work to do things on the inside of the cell. So products will be brought in, and then once they're brought in, the endoenzymes inside of the cell will go to work and change them around to what they need to do. Most enzymes that you will find in a biological system are going to be endoenzymes. And that makes sense if you think about the fact that most of our energy and all that stuff is going to be made inside of our cells because we need it on the inside of our cells, not so much on the outside. A couple more enzymes, where they're found, what they do, what their substrates are, and things like that. Okay, so we want to make enough enzyme and make sure that we have enough enzyme present at any given time to do what our cells need to do. And this is the concept of if you go to the gas station and they don't order enough gas, they're not going to be able to supply everybody. So if our cells don't make enough enzymes, we won't be able to do anything when we need them. And if the gas station orders too much gas, it's all going to go bad before everybody can buy it. So if our cells make too much enzyme, it's all going to go to waste. It's not really useful at that point. It's just going to sit there. It costs a lot of energy to make those, and they're not going to do anything. So cells need to regulate how much enzyme they produce and at what point in time they produce it. So there's two different types of enzymes, once again on a really broad level, constitutive and a regulated. So these kind of enzymes are always present. They're produced at equal amounts, at equal rates, regardless if the substrate is present or not. So you can see here, you've got empty substrates, or you got extra substrates that aren't an enzyme, a bunch of extra enzymes. You add some more substrate, all the enzymes are going to be filled. These are going to be things for really common substrates, things that break down glucose, things that break down things that are eaten on a daily basis that the cell comes in contact with all the time. So cell can pretty much hedge its bets that it's going to come in contact with glucose or something like that on a daily basis, so it's worth making these constantly all the time. Now, regulated enzymes, on the other hand, are going to be either turned off or turned on to make more or less of that particular enzyme when you need it. So in this case, you can see here we have a little bit inside of the cell. This is things like lactose. Cells don't break down lactose all the time. They don't really need to. So we need to make lactase. Lactose comes in contact with it. The cell goes, oh, there's a bunch of lactose now. We need to make more lactase. So it turns on the enzyme to do that, make way more of it. And the other way around. Once the lactose is gone, the cell goes, oh, there's no more need to have lactose. It stops making lactase, and all of that enzyme level goes back down to normal. So you can have makes more of it when there's not, or makes more when there's more showing up, or you can have it where it turns it off and makes not any of it, or makes less of it when it goes away. So both of them work either direction. It can be an A to B or a B to A flow. It depends on the type of enzyme and the substrate in question. So these kind of enzymes, always present, always there, equal numbers, equal amounts, very common substrates. These kind of things, not so much common, not so much going to be things you eat daily. So the bacteria and organisms will make these types of enzymes when they need them. Okay, so enzymes, when they work, they're going to either break things apart or put things together. So the two different types of reactions, when we start digging in on this stuff on an enzymatic level, are going to be synthesis or condensation reactions, or hydrolysis reactions. So synthesis reactions, synthesis means putting things together. So you can see here we have our enzyme here. We have two glucose molecules. Those two glucose molecules are going to have a little water molecule in the middle of the condensation part. It means it forms water. So the two HOH groups on either side of those glucose molecules are going to get close to one another. The enzyme is going to shift, cause those two molecules to bond together, expel one of the waters as a byproduct, one of the OHs on this side and one of the O's or H's over here. So you're just left with an oxygen bond, and it's going to expel that water molecule into the environment, to the condensation part here. And then you're going to have an enzyme release that's going to release the maltose, newly formed maltose, into the environment. So it started out with two glucose, released a water, split a water off of them, stuck them together, and formed a brand new molecule called maltose. So condensation or synthesis builds things up. They also call it condensation because it forms water. So this is a condensation reaction. So this is a hydrolysis reaction. Hydrolysis means splitting of water. So this is the other way around. So you're going to take something that's put together, you're going to tear it apart. So you're going to put the water in, to split apart those bonds to form it back to what it was originally. So you can see here we have our peptide bond in the middle that's holding our AA1 and AA2 together. We stick our water back in the middle. It's going to retake its positions that it had originally before the water was split out in an earlier synthesis reaction. The enzyme's going to break apart that molecule after the water stuck back in and release back out the original products. So this is what originally came back in to the enzyme to start with before it was put together. So hydrolysis means splitting of water. Condensation means putting water out, so building of water. So this splits water apart, uses the energy inside of the water and the H2O molecules itself to kind of fill up the bonds and make things happy and get them to split apart. Whereas this one uses the bonds itself, rips the water out to force things to stick together. So both different types of reactions build things up, tear things apart, but they both require an enzyme to help do that. Okay, so enzymes, activity of the enzyme is influenced by the cell's environment, which makes perfect sense. If it's hot outside, cold outside, lots of salt, lots of sugar, lots of water, different things like that all will affect how fast an enzyme works. So they work under certain temperatures. So our bodies run at about 98.6, somewhere around in there. Anything higher than about 105, our enzymes start to cook, kind of like an egg. It denatures the proteins, and they just turn into mush, and they quit working. Anything colder than about 95, 94, they have a really hard time working as well. It's too cold for them, so they don't work quite as well. So it's called their optimum temperature range. Same concept there with the bacteria and their optimum temperature range. So what happens when an enzyme gets changed or when the habitat becomes unstable is they become labile, which means that they become unstable. So a labile enzyme or labile enzyme, depending on how you want to say it, is something that might work, might not work. It's not necessarily going to be the most reliable thing on the planet. It's too cold, too hot, the pH is too acidic or too basic, too salty, not salty enough, things like that. It might not work the way it should. And then one step farther than that is if it gets too hot and the enzyme itself breaks apart, it becomes denatured. Or if the acid is too high, it breaks apart the bonds that hold the enzyme itself together, the protein itself together, and they fall apart, and then the protein just doesn't work. The enzyme fails to keep its shape, and it doesn't work anymore. And then it's called denaturation. Okay, so once again, how cells can and can't turn off enzymes there's lots of different ways, but one of the ways is using something called an inhibitor. And there's two different types of inhibitors. There's a non-competitive inhibitor and a competitive inhibitor. And then I'll come over here and show you guys what that means. So, competitive inhibitors. So this is our active site in the enzyme. A normal substrate comes into our active site on the enzyme, fits perfectly inside, the enzyme does its thing, and the reaction proceeds. Now, a competitive inhibitor is going to come into our active site. You can see it fits perfectly in there. It's not what's supposed to be there, but it fits perfectly. And it's going to fit perfectly enough that the enzyme doesn't know that it's not what it's supposed to have, and the enzyme's going to try its best to do what it needs to do, but it can't because it's been blocked. The active site's blocked, and essentially what's going to happen is this enzyme's going to be turned off until this competitive inhibitor is released, and that can be permanent or a very temporary amount of time. It just depends. So as long as this active site remains blocked, this enzyme can't do anything. It will never release this competitive inhibitor unless it's told to. So this enzyme's permanently turned off. So the cells can regulate this stuff in some circumstances, but not always. Excuse me. The other way is a non-competitive inhibitor. As you can see over here, on the other side, we have our enzyme and our active site here. Normal substrate looks kind of like a dog bone, comes inside, fits perfectly, does its thing. Now, a non-competitive inhibitor sits on a non-competitive site, so the non-active site. These are often called secondary sites or regulation sites. There's a bunch of different names for them. Non-active sites on the side of an enzyme. So essentially, this is putting a door stop behind the door. So even if you stick the key inside, you're not going to be able to get the door open. So what happens is these non-competitive competitive inhibitors, they bond on those non-active sites. But when they do that, it causes the active site to change shape. So this enzyme won't be able to do anything because the substrate can't come in. So the active site's not actually blocked, but it's had its shape changed because of this non-competitive inhibitor. So it's not competing for the active site. It's just still making the enzyme not work. This one over here is competing for the active site, which is why they call it competitive versus non-competitive. So these are pretty cool. Lots of different ways that the cells regulate these. These work in tons of different enzymes, tons of different circumstances, lots of different substrates, lots of different competitive inhibitors and non-competitive inhibitors, tons of them. They all work pretty similar to one another, though. So enzymes can also be regulated by the process of things called operons or DNA regulation, gene regulation, where an enzyme is produced. It produces a substrate that ends up being produced a product. That product is going to essentially turn off the genes that produce that enzyme in the first place. So if you want to dig into this a little farther, this is called the lac operon or operonic concepts. There's also there's tons of operons. So if you want to dig into that and get into the lac operon concept of how this enzyme repression works, super cool. There's also positive repression and negative repression, which is also really cool. Okay, so ATP, the whole point of metabolism. Why are we doing all these enzyme reactions? Why are we changing things, building things up, breaking things down? It's all to get the energy that's found in this molecule right here. This is money to make energy inside of a cell. It starts off with an adenine molecule bonded to a ribulose sugar, and then you've got one phosphate group stuck to it, adenine monophosphate, AMP. ADP, add another phosphate, adenine diphosphate. This is a temporary carrier molecule in between state, ADP here for metabolism. You've probably heard of that one before. And then our last one here is ATP, adenosine triphosphate, our energy-containing molecule. Now each one of these little bonds here, you can see the purple bonds, or pink, whatever color you want to say that is, red maybe. Each time one of those little bonds is broken, you strip off a phosphate group. The energy that's held in that bond is going to be released and transferred from that bond into the enzyme or the cell, whatever happens to have broken that bond. And all that energy can be used to do something. So ATP is very, very, very useful. So enzymes will strip that bond, they'll break that bond, and use the energy that's stored inside that bond to do something inside of them. So you have to constantly replenish ATP to do other things with your enzymes. You have to make it takes money to make money, and ATP is the money for cells. So you want to dig into the metabolism concept here. If you want to, you're more than welcome to. This can get really confusing really quickly if you are trying to walk through this without actually seeing things move around. So dig into our supplemental activities and watch the YouTube videos of watching these things move around. It's a little easier to understand how ATP is formed, with the formation of glucose and things like that. Okay, so you're probably familiar with these things already, how different organisms make energy. You're probably familiar with aerobic respiration. These are organisms that use oxygen to carry out metabolism. So glycolysis does not require oxygen. The Krebs cycle, the electron transport chain, does require oxygen. There you go. That's the big one right there. You're going to have oxygen for organisms that carry out the electron transport chain for the most part. There are other ways to get around this. It just doesn't work quite as well. So organisms that carry out aerobic respiration are going to require large amounts of oxygen in their respiration, in their I want to say diet, but that's not really a good word. So they don't die when they produce ATP. The other type of respiration is anaerobic respiration, and this does not require oxygen. They'll use the electron transport chain in some sort of way, and they'll use something other than oxygen as the final electron acceptor to steal off the electrons at the end of the electron transport chain. The last one is fermentation, which is the use of glycolysis and then breaking down that energy found in and all that glucose did to produce acids or alcoholic compounds and things like that that will eventually take off those electrons. So that's organisms that don't require oxygen, that don't use oxygen, would be like yeast and organisms that produce acids and things like that. So alcohol, vinegar, different products like that produced via fermentation. So these are the different types of ways that organisms can go about producing energy, either with oxygen or without oxygen, and then how they're going to get rid of all the products that's going to be the toxic products that are going to be built up via making energy. So if you want to view over these again, you're more than welcome to dig into our supplemental activity here of aerobic respiration, anaerobic respiration, and fermentation. You can dig around how this stuff really works if you want to. Get into the nitty-gritty and watch the glucose be split into two pyruvate molecules, which will essentially be split into acetyl-CoA and all that fun stuff which goes in the Krebs cycle. You can watch all this stuff go around if you want to in our supplemental activities. So here's the quick rundown of aerobic respiration. Requires oxygen. Glucose is going to be split into two molecules of pyruvic acid, NADPH, or NADH, excuse me, is going to be generated. It's a temporary carrier molecule for energy, super useful. We'll talk about that later. You're going to have the Krebs cycle, or carbon reactions. It depends, lots of different things call them different things. Which is going to take those two pyruvate molecules that are coming in from here, generate 3CO2 that we're going to breathe out, NADH, and two FADHs. You can see those over here. The whole time we're stripping away electrons off of different molecules as well, sticking them inside of our mitochondria that are going to be used to power the ATP synthase pump, which we'll talk about in just a second. The very last step is to take all of those electrons that have been stripped out into the electron transport chain to move over all the NADH, FADH electrons over here. And then we're essentially going to run them through a bunch of different reactions, all those different electrons, to strip the energy off of them to power other things, which is essentially going to make ATP. Really long, really complicated reactions. Dig around in our supplemental activities if you want to get the nitty-gritty of this one. Essentially, aerobic respiration is going to generate around 38 ATP. Realistically, it's more like 25 to 26 of them. But in theory, it can make 38. It takes energy to make energy, so you're not going to get all 38 out of it. It takes energy to do this as well, and it doesn't always work as well as it should. So glycolysis, dig into our supplemental activities if you want that one. Say with pyruvate. Krebs cycle, dig into that stuff. The electron transport chain, dig into it if you want to on our supplemental activities. Okay, so here's our ATP synthase molecule. Essentially what happens is all of the enzymes have been broken, or all of the electrons have been broken apart. All of the hydrogen molecules have been stripped apart. All of the stuff that we want has been done at this point. And this is our bacteria. This is the cell wall here, in the periplasmic space in between the cell membrane and the cell wall, gram-positive cell. Little space that sits in between is going to have tons of hydrogen molecules stuck in between it. We've stripped them off of all of the other FADHs and ADPs, all that stuff. So we're going to get all these H's and all these electrons. So all the FADHs and stuff we talked about earlier. So all those H's that have been pumped up there are going to cause a concentration gradient. Now what's going to happen is we're going to constantly pump more of those H's and stuff from the cell that we strip off of the NADPH on the inside into that periplasmic space. This takes energy. The energy that is going to provide this is going to be power. This is going to be provided from the electrons that we stole earlier from all those other molecules. So the H's are going to come up to the cell membrane, be pumped across via the little cells channel proteins, that sit on the cell membrane. The electrons are going to power that. Pump a ton of hydrogens on one side, not very many on the other, ton on the other. This causes a concentration gradient. Now what's going to happen is this big giant molecule here is called ATP synthase. It works kind of like a propeller. ADP from earlier that had the ATP turns into ADP when you strip off the second P. sorry, the third P. So that ADP molecule that had the third P stripped off of it is going to come up very close to this ATP synthase molecule. A phosphate molecule that was stripped off earlier from the ADP to make or from the ATP to make ADP is also going to come up close. One of those little hydrogen molecules is going to be stripped down through that ATP synthase molecule just because of the concentration gradient concept of diffusion. High concentration, low concentration. The hydrogen is going to be ripped down real quickly through that ATP synthase molecule. When it does that, it's going to spin it. Oops, sorry. Kind of like a went the wrong direction. It's going to spin it like a boat motor, like a propeller. And when it does that, it's going to sling that ADP and that ATP or that phosphate group together really quickly and force them together to form ATP. And that's how this works. That's why we need to strip all those hydrogens. That's why we need to strip all those electrons as well. Now, in your cells, human cells, this happens in the mitochondria. In bacteria, prokaryotes, this happens in their cell wall, in between their cell wall and their cell membrane, that periplasmic space. Because they essentially are just a giant mitochondria. So oxygen is going to be used to get rid of all the extra hydrogens here. We're going to breathe it out in the form of water, essentially water vapor. And that's what the point of getting out all those oxygens. That's why you need oxygen in your metabolism. So oxygen is going to be stripped away. You can see here, it's going to pick up two of those hydrogen molecules, and you're going to release all those extra hydrogens as water back into the environment. So once the hydrogen's used, you just get rid of it in the form of water, and that's why oxygen is so important. So you have no oxygen, you can't get rid of your hydrogens, this process backs up, you can't form any energy anymore, and you die. So that's why oxygen is very important for organisms like us. So here's how that one works. You can see which step produces what. ATPs are produced at a couple of different steps, but not many. And the big step down here, our electron transport chain, is going to produce the most. So nice little quick breakdown of what's produced at what step, and how many ATPs are theoretically produced at each step as well. So pretty cool. So here's anaerobic respiration, the opposite of this one. Doesn't require oxygen, doesn't use oxygen as our terminal electron acceptor. It uses other things like sulfates or carbonates or nitrates and things like that to steal off those hydrogens at the very end. So it'll produce a hydrogen sulfate or a hydrogen carbonate and things like that, hydrocarbons and stuff at the very end. So very interesting concept, still very useful to produce energy, but not quite as effective. So you can dig on that one if you want to, how that one works. So here's fermentation. You can produce different types of things with fermentation. Sorry, I skipped a little far ahead. Different types of organisms produce different products via fermentation. If you're a prokaryote, you are going to produce, for the most part, acids. So you're going to produce lactic acids, different types of acid products like vinegar, things like that, acetic acid. So that's what most prokaryotes produce. On the other side of that, eukaryotes, for the most part, other than human muscle cells, which produce lactic acid, almost all other prokaryotes, or eukaryotes, if they have to undergo fermentation, this occurs when you run out of oxygen. This is in human cells, the kind of last-ditch effort of, oh my gosh, we're about to get eaten by the bear, we better run away. Let's give it a last little ditch of effort. But for other organisms, like yeast and things like that are eukaryotes, they can undergo fermentation just kind of naturally. And when they do that, they produce alcohol. So that's the fun product of their metabolism via fermentation. So prokaryotes produce acid. Eukaryotes, other than human muscle cells, which produce lactic acid, will produce alcohol. So fun little thing there. So here's how this one works. You kind of see what's broken down. Glucose is our start here, what breaks it down, and what's formed from each little system here. So metabolism is super cool because things that you eat can be used to power different cycles. And this makes sense if you think about it. you eat different types of things, different types of macromolecules on a daily basis, and you might not necessarily eat a balanced diet. So your body evolves ways to deal with different things that you might eat, and to use them in ways that they might not necessarily be meant to be used for the most part. So if you eat a lot of proteins, your body's going to use the vast majority of them to make amino acids. If you eat a balanced diet, it doesn't really need to use proteins to make anything else because it has everything else it needs. But if you eat a diet that's high in proteins and low in everything else, your body can use those excess amino acids to make other things in your body to power metabolism that you're not getting, because you're not eating a necessarily balanced diet. Now, it doesn't work quite as well, but it does work. So it's kind of a backup system that your body can use to make other things to power metabolism when you're not necessarily eating or getting all of the nutrients that you need. So super cool way that our bodies have evolved and other organisms have evolved to get around the process of living. So photosynthesis, super big deal in metabolism, process of taking light energy and taking that light energy plus some carbon dioxide and water, and turning that into glucose and oxygen. So the plants will use that glucose for them to eventually break apart to make ATP with. So they run it through that same metabolism process. They just have an extra step to make it. So they make the ATP from power of photosynthesis, and then it runs through the exact same processes that we just ran over with for them to make ATP. They just have that extra step involved to make their own glucose. We get our glucose from eating it. So extra step involved with them, but same process. So there's two different steps of photosynthesis. There's the light-dependent reactions and the light-independent reactions. It really just depends on the book if they call them the light or the dark reactions or the light-dependent and light-independent reactions. But our light-dependent reactions are going to occur when photons are going to be absorbed by chlorophyll. The energy from those four photons are going to be stored and then used to strip apart water molecules by photolysis, which means stripping water via light, releasing the oxygen gas stored within those water molecules, stripping out the hydrogens, things like that. So essentially what's going to happen is the light energy that's going to be released and stored by all those photons is going to be captured, from the photons, and then eventually released is going to be used to synthesize ATP and NADPH, which will then be used later. So our light-independent reactions, sometimes called the dark reactions or the Calvin cycle, is then going to use ATP to fix carbon dioxide and a fun enzyme called ribulose. It's the most abundant enzyme on the planet. Every single plant has billions of this stuff inside of it, and make it into glucose. So then that glucose is going to be used to carry out photosynthesis or carry out metabolism inside of a plant cell. So dig a little deeper on this one if you want to, on our supplemental activities, on how plants make energy if you'd like. But other than that, that's really all I've got for you guys on this one. This is a super brief run through on metabolism. I'm not going to dig super deep into this one. This is meant to be a really broad overview, really quick kind of down and dirty introduction to enzymes, kind of how they function, kind of their role in metabolism, and a little quick rundown on over the different steps of metabolism and things like that. So if you guys have any questions, shoot me an email. If you want to have a discussion or anything about this, and if you need a little extra information or help, feel free to let me know, and I'll be happy to schedule a Zoom session or anything with you guys. Other than that, have a great rest of your day.